Dear students, today we are going to talk about splicing machinery. Splicing machinery. This splicing machinery, which is known as spliceosome, it mediates splicing. It mediates splicing. It contains this spliceosome. It is a complex. It's a Spliceosome is a complex. It is known as spliceosome complex. It contains 5 RNAs, 5 types of RNAs, and almost 150 proteins. Spliceosome complex, it is a splicing machinery. It helps in joining exons and removing introns from the pre-messenger RNA. It contains 5 RNAs and 150 proteins. Okay, as these RNAs, these RNAs are called snRNPs. snRNPs, small nuclear RNAs. Okay, and some 150 proteins. These snRNAs are usually U1, U2, U4, U5, and U6. And U6. These are the 5 types of 5 RNAs. These snRNAs, U1, U2, U4, U5, and U6. Here, U3 is not involved. There is no U3 in spliceosome complex. U1, U2, U4, U5, and U6 are the 5 types of RNAs involved in splicing. Okay, these U1, U2, U4, U5, U6 RNAs, they are, as we know, RNA is made up of nucleotides. These RNAs are made up of 100 to 150 nucleotides. It is the range of nucleotides present in these types of snRNAs. As we know, in spliceosome, snRNA is complexed with some 100 to 150 proteins, snRNAs plus proteins. snRNAs are complexed with proteins. They are called small nuclear ribonuclear proteins. Small nuclear ribonuclear proteins, or they are called snRPs. snRNA complexed with proteins are called small nuclear ribonuclear proteins, or they are also known as snRPs. S here small, it says nuclear, N ribo, nuclear NU ribo, this, and proteins. So hence they are also called small nuclear proteins. For the arrangement of spliceosome, it needs ATP. It needs ATP. For the arrangement and for the proper functioning of spliceosome complex, it utilizes the energy by hydrolyzing ATP. Okay. So what does this spliceosome complex do? What is the function of spliceosome? As we know, spliceosome, it removes the introns and ligates or joins the exons. Okay. Usually snRNPs, snRNPs, they perform three important functions. Okay. So what are these important functions? First of all, we will draw structure of pre-mRNA. Here, for example, this is an exon. In between, there is an intron. Okay, as in previous lecture, we have talked about this. This is 5' site and this is 3' site. Here, at the junction, this junction which is in between in this exon and intron, this is exon, this is intron, this is exon. This site here, which is the junction between exon and intron, here it is guanine and uracil. It is present at the terminal of, at the 5' terminal of intron. This GU site, it is present at the 5' terminal of the intron. Okay, this site is called splice, 5' splice site. 5' splice site. Okay, these are the some nucleotides which are important for splicing. Here, an important nucleotide is present. It is called a branch point. This site, it is called a branch point. And at the 3' terminal of the intron, here it is adenine and guanine. This 3' terminal site of the intron is called 3' splice site. These three sites are important for splicing. As these sites are important, they contain consensus sequences GU, A and AG. These are the consensus sequences within the intron. Okay, GU site or GU nucleotides, these are present at the 5' terminal of the intron, and AG is at the 3' terminal of the intron. This A site, A nucleotide, it's within the intron, but it is more close to 3' terminal site than 5' terminal site. It's called a branch point. Okay, now what is the importance of these sites? As we are talking about the functions, three important functions performed by the snRNPs. First of all, snRNPs, they recognize the 5' splice site. 5' splice site. Okay. snRNPs, they recognize 5' splice site and branch point. So these sequences are important for the recognition by spliceosome complex. 5' splice site and branch point are recognized by the snRNPs. Okay. Then second function, which is performed by the snRNPs, they bring these sites together. They bring these sites together. Okay. And the third important function is they catalyze the RNA cleavage, and joining reactions. They catalyze the cleavage reactions and the joining reactions. Okay, these are the three important functions performed by the snRNPs. Number one is 5' splice site and branch point are recognized by snRNPs, and then they bring these sites together. And third function is they catalyze the RNA cleavage and joining reactions. Okay, okay, this GU site, which is called 5' splice site, it is recognized by U1 snRNP, U1 snRNP, and it is also recognized by U6 snRNP. Okay, 5' splice site, it is recognized by U1 and U6. Okay, this branch point, it is recognized by U2 snRNP. U2 snRNP. Okay, it's also recognized by a protein which is called branch point binding protein. Okay, this branch point is recognized by branch point binding protein, and it is also recognized by U2. Okay, this, first of all, this branch point binding protein, it comes and it binds to this branch point. Then U2 replaces this branch point and gets attached to the branch point site. Okay, we can say that this branch point binding protein, it recruits U2 to the branch point. Okay, then U2 snRNP and U6 snRNP, they show U2 and U6. Then they show RNA-RNA pairing. They show RNA-RNA pairing. U6 and U2. They show RNA-RNA complementary pairing. And they bring 5' splice site and branch point together. Branch point together. Okay. Now in the next frame, we will show what is the mechanism of spliceosome machinery, or we can say what is the mechanism of splicing pathway. Okay. Now we will talk about splicing pathway, how splicing occurs with the help of spliceosome complex. Okay. Here it is pre-messenger RNA. It's pre-mRNA. Okay. This is 5' site. This is the 3' terminal. 5' terminal and 3' terminal. This is an exon, and this is intron. Okay. First of all, at the 5' splice site, U1 snRNP, it gets attached to the 5' splice site. Okay. Then at this site, which is called branch point site, it contains adenosine, comes a spliceosome protein which is called branch point binding protein. It gets attached to this site which is called branch point site. Okay, this complex is called E complex. Okay, then in the next step, this branch point binding protein, it gets detached. It recruits U2 snRNP. Okay, So what happens? A complex is formed. Okay, this 5' splice site, here it is U1, and now at this site, at the branch point site, where it is adenosine, branch point binding protein, it recruits U2. So U2 gets attached to it. This U2 this U2 snRNP, here U2 gets attached and it's long. It occupies branch point as well as 3 splice site, and it also extrude this adenosine. Okay, it extrudes this adenosine from the frame. Okay, now it comes here. So extrusion of adenosine occurs with the help of U2. Now this adenosine is ready to react with the 5' splice site. It's now ready to react with 5' splice site. Okay, now it acts as nucleophile. This complex is called A complex. This is called A complex. This was A complex, and now this is A complex. Okay, in the next step, what happens? We'll show here. After A complex, next step occurs. 3 snRNPs in a trimer shape, they comes and bind with the pre-messenger RNA. These are U6, U4, U6, U4, and U5. U6, U4, and U5. Okay. Here I will show another complex after the binding of U6, U4, U5. What happens? This U6, U4, U5, it comes in a form of complex which is called tri-snRNP complex. Tri-snRNP complex. Okay. Here we know it is the adenosine which is extruded. These U1, as we know, it is already binded to 5' splice site. Then comes U6. As we know, U1 and U6 complementary site at the 5' splice site. Then U4, then U5, and here it is U2. This complex is called B complex. This is called B complex. Okay. This complex, when U6, U4, U5 comes, they bring these sites together. They form a loop-like structure here. Okay. Now in the next step, U1 is released. U1 is released, and U6 binds. It comes and it joins. It forms the complementary base pairing with the 5' splice site U6. Okay, we have U4, we have U5 and here A with U2. This complex then what happens, this U4 is removed. U4 is removed. U4 is removed and five prime splice site, it is cutted, and this 5 prime splice site is joined with the branch point, okay, forming a complex which is called C complex. This is called C complex. Here we have U6, U5 and U2. Okay, this U6 and U2, they interact with each other, okay, bringing 5 prime splice site close to the adenosine, forming a lariat like structure. This complex is called a C complex. Then another transesterification reaction occurs which brings this site, this 5 prime exon, close to the 3 prime exon, okay, and forming the complete mature messenger RNA. Okay, now this lariat comes out. Okay, so this was regarding splicing pathway. In the second esterification reaction, I will show here. I shall rub it. Okay, U5 snRNP is involved in the second esterification reaction. U5 snRNP is involved in the second esterification reaction. Okay, it brings the two exons close to each other, and final step mRNA is released. mRNA which is mature mRNA, it is released, and snRNPs are also released. There is a protein which helps in the stripping of the spliced mRNA from the spliceosome, that is called dead box helicase. Dead box helicase. There are different dead box helicases. One of the important is PRP22. This protein is required for the second catalytic step of splicing, and also PRP22, it is also used for stripping the spliced mRNA from the spliceosome. So it was about spliceosome complex and splicing pathway. Thank you. In the next lecture, we will talk about what are the types of introns. Okay, so that will be discussed in the next lecture. Thank you very much.